Welcome to Biology Made Easy. In today's lesson, we will look at monohybrid inheritance or cross. Mendel brought out monohybrid inheritance and dihybrid inheritance. So why was Mendel so successful? Is because he used specimens with contrasting characteristics. For example, cowpea seeds, he looked at skin texture and contrasting smooth and wrinkle, two contrasting characteristics of a gene. The gene is skin texture. Mendel was also very successful because he used small size specimen, so he could grow plenty of plants in a small space. He also used materials with short life cycle, so the generation produces results quickly and another generation comes in. That is Mendel. And one of Mendel's experiments is on monohybrid cross. Monohybrid cross is a cross or mating that involves two individuals where one gene is considered and the gene has a pair of contrasting characteristics. The characteristics do not merge at all. For instance, we can have a gene for height with contrasting characteristics tall, dwarf plants. That is monohybrid cross. Now, these are some of the simple experiments that is done. Tall plants crossed with dwarf plants. The tall plants, let's say they are the males, and the female are the dwarf plants. Now, the cross involves picking pollen grains from one plant and putting it on the stigma of another. So pollen grains from the tall plants and putting it on the stigma of the dwarf plants. You see, a polythene bag is tied around so that no other pollen comes around. So this fruits, the seeds are germinated and grow, and all the plants that were formed were tall plants. So these plants are the F1 generation. This is the F1, all tall plants. So the F1 plants, the tall plants, were celled. It means self-pollinated. Pollen grains were picked from the anther and put on the stigma of the same plant. And then fruits are formed, seeds are germinated, they grow. And what was the result? The results produced tall plants as well as dwarf plants. 3 out of 4 of the plants were tall, 1 out of the 4 dwarf. And these plants that were produced were the F2 generation. We explained all these terms in our last lesson. We'll use genetic diagrams to explain this process. And in this process, you see we had tall plants and dwarf plants mated and produce all tall plants. It means that tall is dominant, and it also means that tall is pure breed, that is homozygous dominant. So tall is dominant, dwarf is recessive. So the genetic diagrams are these. You have parent phenotypes, tall plant crossing dwarf plant. Parent genotypes, 2N, capital T, that's a homozygous dominant pure breed. Meiosis takes place in the anther of the male plant. Meiosis takes place in the ovule. So meiosis takes place, we explained all these in our last 2 lessons. So meiosis, and then gametes are formed. You see this parent is big T. All the gametes that will be formed will have only big T, and the gametes are haploid. 4 gametes would have produced from each, but look at it, because the 4 gametes are the same, we write 2 of them. And here too, these are the gametes that will be produced, small t. And from this, we would have had 4 gametes. But because they are the same, we write out only 2. Random fertilization takes place, and random fertilization, this gamete can cross with this gamete. So that is it here. And you have this individual form. This same gamete can cross with this gamete and have this. That is why we didn't write all the 4. So this one can do it 2 times, this here and the same thing here. This one can cross with this to have this. This same one can cross also with this to have. So this is what we have F1 genotypes. They are all big T, small t, and the phenotypes are all tall plants because tall is dominant over dwarf. So the phenotype are heterozygous tall. That's what happened in this situation here. Now, selfing the F1 means here you pick pollen grains from here and put it on the stigma here. And that is how the genetic diagrams explains it here. Selfing. So we have tall parent here crossing tall. That is the selfing that is done here. The genotypes is big T, small t, because that is it we use here. This is the genotype. And the genotype tall here. Meiosis takes place and you have the gametes form. You have one gamete like this, another gamete like this. And there will be 4. So they would have been big T 2, small t 2. But because it's the same, we wrote only one of them, so that we can cross 2 times. Okay, so we have big T, small t, so you have gamete big T, gamete small t, because the gametes separate. We talked about it in the meiosis process. So random fertilization will take place. This gamete can cross with this one. A big T, the same gamete can cross with this one and have big T, small t. This gamete can cross with this and have big T, small t. This gamete can cross with this and have small t, small t. So F2 genotypes will be this, 2N, and F2 phenotypes, this will be tall because tall is dominant over dwarf. So this is tall, this is also tall, and this is dwarf. This is the F2. F2 here, we had here, is 3 out of 4 is tall, 1 out of 4 is dwarf. So look at it, 1, 2, 3 is tall, this one is dwarf. So we are going to use the punnet square to show combinations of gametes. So this is gametes that is put into the punnet square. What we do is to draw like this, put in this, put a cross here. We have the male gametes. They are going to be on this line. The female gametes are going to be here. So this is the male gamete. So we have big T here, small t here. This female, we have big T here, small t here. These are the gametes. So we write here gametes here and here. So random fertilization takes place. You have this female gamete will fuse with this male gamete and have big T, big T. This one will fuse with this and have big T, small t. This will fuse with this gamete. You have big T, small t. This will fuse the gamete to have small t, small t. So we have these as the offsprings. We have this is tall, this is tall, this is tall, this is dwarf. So you see we have 3 talls, 1 dwarf. So the punnet square can be used to get the same results as these crosses. And the punnet square tend to be easy, as we can see later on. Good. So you see the ratio 3:1 is known as the monohybrid ratio. And it's 3:1. The monohybrid ratios can also be explained in probabilities. So you have this parent genotypes, big T, small t, crossing big T, small t, as we had here, and you have the gametes. Now, this big T, small t, the probability of a gamete being big T is 50%, half. The probability of a gamete being a small t is also half, 50% half, 50% half. So we write the gametes down and the probability of the gamete is half, probability of this gamete is half, half, half. Random fertilization takes place. This gamete can fuse with this gamete. You have big T, so half times half, you get 1/4. So this gamete will also This gamete can also fuse with this gamete. You have big T, small t here. This gamete is half, this gamete is half. Half times half is 1/4. The same thing happens with this gamete and that gamete. Half times half, 1/4. Half times half is 1/4. So when you put it all together, we have 3/4 talls, 1/4 dwarf. We come to the same monohybrid ratio of 3:1. That is the monohybrid ratio using probabilities. Now we want to consider test cross. A test cross is a cross to find out whether a heterozygous which shows a dominant character is homozygous dominant or not. So you cross the individual with a dominant character with a homozygous recessive. These F2 individuals we have here, we have big T is tall, big T, small t is tall. They are all tall. So we don't know whether phenotypically they are all seen as tall. But one does not know whether it's a tall homozygous character or a tall heterozygous character. So a test cross is done to cross the individual who is genotype you don't know, but it has a dominant character. The dominant is the tall. This individual is crossed with a homozygous recessive. This is a homozygous recessive because tall can be big T or big T, small t. If tall plants are homozygous, then we have tall plants, and in the test cross, we are crossing tall plants with homozygous dwarf plants. This is the parental tall against dwarf, big T, small t. Gametes will be all this, all that. Random fertilization will produce big T, small t, or big T, small t. So all tall plants, if the tall plants are homozygous tall. If the tall plants are heterozygous tall, then we'll have the parent tall against the homozygous recessive that we are using as a test cross. We have big T, small t. This is a heterozygous. These are the gametes that are produced, and this one will fertilize this together. So we have these plants as tall and these as dwarf. So if the tall plants were heterozygous, half of the offspring will be tall and half will be dwarf. This is a test cross. Well, this is what we have for today. If the lessons have been useful, please give us your thumb and do subscribe. Thank you very much. Bye-bye.